it off to you, Hoifung. Thank you again.
Awesome. Thanks, Susan. I'm very excited to be here. And I will be telling you guys a little bit about our recent experience in developing state-of-the-art biomedical natural language processing to help in medicine. So I will first introduce precision medicine and explain a little bit why we think that NLP and machine reading can potentially help here. I will then review some of the difficult machine learning challenges and a couple of the research frontier of self-supervision and novel neural architecture. I will conclude with some exciting end-to-end application scenarios. So in the ideal world, we want, obviously, every patient to be able to respond to the treatment they are prescribed, as signified by the blue person on the left here. In the real world, unfortunately, most of the patients don't respond to the treatment as represented by the red person here. So the challenge used to be that we don't have a lot of the fine-grained information to be able to distinguish the blue from the red at priori. So the advent of big data in medicine promised a change for the good, right? So cancer is a poster trial for this. By cancer biology, we know that the tumor is driven primarily by the mutation. So with the tumor genome, we are literally staring at the sort of the source code for the cancer and can potentially distinguish the patient at the individual level. Another really exciting sort of disruption, as the two mentioned in the first session, is the rapid digitization of medical records, right? So those provide a lot of fine-grained longitudinal patient information. However, the big data also present this grand challenge of information overload, because the big data by themselves is not very useful unless we can actually able to interpret them and translate them into actionable treatment insight, right? Right now, this is done completely manually, which is very hard to scale. For example, a lot of those knowledge that are required to interpret the tumor genome reside in the literature, which add thousands of papers every day and over 1 million a year. So even curating a single paper can easily take hours. Similarly, we may want to find a lot of sort of like patient like me information from the electronic medical record, but the bulk of the information there often can only be found in text consistently, and abstracting medical records for a single patient could also easily take hours. So this has motivated our research agenda in Project Hanover, where our overarching goal is to develop machine reading technology to help unlock all those key information from medical texts. For example, a paper might start by describing this patient cohort with this certain gene and mutation, and then went on to report that the patient was treated with a certain drug and showed partial response. Now, from this piece of text, we want the machine reader to be able to extract a potential finding that the tumor with this gene and mutation may be may be sensitive to this given drug. So from the literature, the overarching goal, obviously, the ultimate goal is to extract this PubMed scale knowledge graph, where the nodes are important medical entities such as a drug, gene, mutation, disease, etc., right? And then the edge or the hyper edge represent binary or complex relations such as drug response or gene network. From the EMR, as you heard Atu mention in the first session, a particularly exciting direction is to collect all this sort of population level real-world evidence of very fine-grained longitudinal patient treatment trajectory from the diagnosis to treatment and the outcome. So all these are very exciting, but machine reading can be very challenging. First of all, authors like all of you could be very creative in figuring out a different way to say essentially the same thing, right? So here are just a few ways to say that the tumor suppressor can inhibit the apoptosis pathway. Now, if you just look at a couple small corpus of a couple dozen PubMed abstracts, you will already see an extremely long tail of words that have been used to signify this kind of negative regulations. Now, conversely, same expression can also mean completely different things, right? So there is a gene actually called PDF, but if you go to PubMed, most of the PDF mentioned doesn't refer to the gene. So the machine reader will really have to strike hard from the context to figure out whether a particular mention is a gene reference or not. So far now, variation and ambiguity, they are just pretty bread and butter, NLP challenges. But in biomedicine, we are also forced to tackle additional challenges that are often underlooked in the mainstream NLP, which focus on newswire and the web, right? So for example, the majority of the information extraction or machine reading work have been focusing on simple binary relation that occur within a single sentences. But in biomedicine, we need to extract all this complex, potentially N-ary relations, such as this precision oncology knowledge involving drug gene mutation. And you can also easily ask about what about cancer type, evidence type, etc. And the entity involved in such a complex relation can often span across sentences or even paragraphs, right? So conventional, NLP method can easily break down in such a long span complex relation. For example, if you try to run a BER or LSTM over such a long text span, they will not work. Another sort of big challenge is that when we go to biomedicine, we actually lost a very potent tool, which is the crowdsourcing, which is actually behind sort of like fueling a lot of the latest progress in deep learning. So imagining you try to ask the Turker to interpret your genetic paper, or actually figuring out what's wrong in a CT scan, that will be very hard. So that actually motivates us to start looking into a new sort of learning paradigm called the self-supervision. So the key idea is actually very simple, right? So the standard supervised learning, essentially counting on domain experts like you guys who are able to label input-output examples so that you can feed into the machine learning algorithm to train the model. But that labeling process become the bottleneck. So the idea of self-supervision is to leverage all those sort of like free lunch, like those resources that are freely available, to try to come out with something to actually bootstrap the machine learning system, right? So there are two types of self-supervised learning. One type that many of you are probably quite familiar with is that by just focusing on just leveraging the unlabeled text side to try to learn a very good representation. So this, traditional would be all those, early days embedding method or clustering method, but also more recently on learning large neural language models such as BER and Elmo, and I will come back to that later in the talk. Here I will first start by focusing on another type of self-supervision, which is the task-specific self-supervision. And the key idea here is that we can leverage those domain knowledge, freely available domain knowledge like ontology or manually curated knowledge base, to essentially hallucinate a lot of noisy label example in the unlabeled text. So let me, so in Hanover, we have been actually using this paradigm very successfully into a swath of biomedical application, starting from early days, focusing on fundamental biology, such as gene pathways and so forth, and more recently on the translation scenarios, such as the tumor board. So let me start with a very simple example to illustrate the key idea. So for anything that you care about in biomedicine, chances are somebody have tried to manually curate them, right? So often you got a grant, you assemble a team to curate, you've got a pretty good knowledge base, but the grant money runs out and you stop the curation. So the knowledge base often have very poor coverage and also quickly go out of date. But so that's quite unfortunate, but we can try to turn those knowledge bases into sort of like a recipe to actually create noisy label examples. And so if we know a particular fact in the knowledge base, right, like for example, our friends of P53 inhibiting BCL2, it is not a label example by itself. But if we go to the unlabeled text and we realize that, well, these two entities actually happen to co-occur in these sentences, we can now take a leap of faith to pretend that, okay, this is probably a mention of this given relation of interest. And in this way, you can automatically annotate a very large number of label examples for free, right? Now, obviously, you may have a question that, well, the two entities could also have another million reasons why they could occur in the text, right? But one thing to keep in mind is that statistical learning actually have this built-in capability to withstand some noise, some level of noise. So as long as your signal to noise ratio is outweigh the noise, then you can potentially bootstrap the learning successfully. And moreover, you don't have to sort of stop at the simplest self-supervision, but you can also incorporate other type of domain and linguistic knowledge. And probabilistic logic is the sort of a very well-suited framework to incorporate all this diverse and potentially noisy self-supervision. The key idea here is that let's try to model all those latent labels that you wish to know. For example, the Z1 is like whether the first PDF is a gene reference or not. The Z2 is whether the second PDF is a gene reference and so on, right? And we can now introduce the prior knowledge as probabilistic factors like F and G, which collectively define a probabilistic distribution over the latent label variables. And then we can use state-of-the-art statistical inference algorithm to compute their marginal probability, and then pretend they are the probabilistic labels. And from then on, it's pretty straightforward. You can train your favorite deep learning models. So in this way, we introduce this deep probabilistic logic, which is a general framework that allows you to combine all those self-supervision to get around the labeling annotation bottleneck. We can also sort of innovate on the new architecture front. For example, our group is one of the first who introduced a graph neural network into NLP applications, right, by proposing the graph LSTM. So the key idea is that we do have very good syntactic parsers that can give you sort of this kind of dependency tree among sort of like words inside sentences. And we can essentially using those linguistic resources to impose hard attention, right, to help model this kind of very long range dependencies. And we can also, to tackle the document level relation extraction, right, we also propose this multiscale representation learning, where the key idea is that when you have all these entities scattered across paragraph, what you can do is to essentially try to learn a local representation of whatever partial, co-occurrence in the local linguistic unit, like a sentence or a paragraph, and then try to essentially combine them in a sort of a pooling process into a document level entity representation, which you can then use in the final prediction. And also get back to the task agnostic self-supervision, right? We are also actively working on how can we sort of portray this large neural language model so that they can be most effective for biomedical application. So a conventional wisdom is to start from a general domain, a BERT model that is trained on the web text, right? And then try to continue pre-training using biomedical text. What we find is that actually these assumptions appear to be wrong because in biomedicine, we already have tons of in-domain text. So by actually focusing our pre-training solely from the biomedical text, we were able to actually outperform all the prior BERT model in a large number of biomedical NLP applications. And to facilitate this investigation, we also created this very comprehensive biomedical NLP benchmark that we call BLURB, And we are going to release leaderboard as well as all this state-of-the-art bird model to help the community to accelerate progress. We have also created a PubMed scale knowledge graph for COVID, and we are working with collaborators to incorporate those information into the number. So all this are sort of pretty promising, but none of this matter until we can actually sort of get some potential end-to-end impact, right? Here, what we think we actually envision sort of the sweet spot for AI for medicine is actually in this sort of like empowering this sort of like human-computer symbiosis. So despite all the hype in AI, right, human biologies and medicine are very complex, and even the most sophisticated AI can be actually rather primitive in medical applications. So for curation in particular, we don't envision that NLP can sort of be able to automate this thing end-to-end with 100% accuracy anytime soon, right? But instead, what NLP can potentially do is to play the foot soldier for human expert by reducing those haystack, for example, the 4,000 paper for today, right, into a few, candidate extractions, and then the human expert can very quickly validate them. So for the tumor board, for precision oncology knowledge, we have created a UI where people can actually look at sort of like the rationale for the machine reader for each candidate extraction, and then they can see them with the provenance and can actually quickly verify them by clicking a button. And we have been working with Cancer Center, the Jackson Lab. They have a team to manually curate this kind of precision oncology knowledge. For a typical drug, the manual curation process will often identify, hundreds of papers that with potentially relevant findings, which normally would take thousands of hours, person hours, to actually curate them all, right? With Hanover machine reading, we were able to summarize those papers, right, in an instance, and produce, for example, in this case, a 40-ish candidate finding, which the JAX curator using our UI was able to curate them, verify them within an hour, and already produce sort of an order of magnitude more findings compared to what they have ever curated in the past. So this is very exciting. So we are writing a paper to report these findings, and we are also working to incorporate the machine reading into their end-to-end curation workflow. Now, going forward, as Atul mentioned, real-world evidence is a very exciting frontier, and we are increasingly sort of focusing on these directions. We have made some really sort of like initial very promising progress on cancer registry in collaboration with a couple other cancer centers, and we are writing up papers about them and hopefully start some test deployment soon. Down the road, we are most excited about some of the really big prizes, such as the virtual trial, right? So, and as you all know, an average FDA-approved drug can cost billions to develop, and a huge cost is in trying to figure out those sufficient statistics among the control arm for patients who are undergoing the standard of care. So companies like FightIron have already shown that you can potentially gather those sort of comparable information from the EMR, but right now they are basically doing this by essentially completely manually with hundreds of abstractors, which is very hard to scale. So if we can sort of replicate our success in the tumor board or the cancer registry, we can potentially have hopefully a very big impact here. So I will end here. So these are the team who actually do the real work, right? I also want to sort of thank our collaborator over the years who has inspired us and in some cases collaborating with us directly, without whom we obviously can't make any of this work. So thank you, and I'm looking forward to discussing and learning from all of you.
Thank you so much, Hoifung. Of course, if you would like to ask a question, the question answer box is freely available. one of the things I'll ask really quickly before we move on, one of the things that we work on pretty diligently, certainly at NIH, is the construction of common data models, common data elements. Do you feel like the work you're doing here in machine reading of text and data, metadata files could help in any way with our wrangling of these sometimes thousands of data elements?
Yeah, I think the two can potentially sort of like help each other and also complementary in some way. So a lot of our machine reading requires sort of like a specification of sort of like the ontology, right? So we're assuming that you already know what are the data element that you want to extract. And so that's sort of like usually is the prerequisite. But there are also NLP methods that we can actually try to generate like what are some of the key phrases, what are some of the cluster of expression from which you can potentially, for example, like social determinant, there is no good ontology, and we have been thinking, hey, can we actually come up with some sort of like potential candidate elements, right? Yeah, love to learn more about your use case and see whether we could help.
Absolutely, that's wonderful. Thank you. And thank you again